This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1215, recorded on the 2nd of May, 2025. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from College Station, Texas, Jolene Ramsey.
Howdy. Good to be here.
I have to think of all of my Texas places. Well, I only have two, right? There are two people in Texas that do podcasts, and they're both on TWiV, College Station. So you're not frequent enough for me to know it by heart. You know, I have to kind of think a little bit, a second or two, but I got it.
Well,
also joining us, yes, go ahead.
Well, here in College Station, we have weather that's very similar to the other place in Texas that records. So today, it has been a nice spring stormy day. It's sunny outside right now, but we've been having sporadic thunderstorms. We had an incredible lightning storm last night, and all the wildflowers are still blooming. So it's lovely weather here.
Here in Chelsea, it is 28 Celsius, which is 82 degrees Fahrenheit. Mostly sunny. It's a very nice day, but I wouldn't know it. I've been inside all day. Also joining us from Western Massachusetts, Alan Dove.
Good to be here. It's 72 Fahrenheit, 22 C here, but humid, 66%. And we also had thunderstorms roll through last night. I was sleeping soundly and was awakened by a thunderclap. Oh, wow. So then this morning it was just kind of cloudy and a little muggy all day.
Thunderclap.
Yes.
Thunderclap. If you enjoy these science programs, we'd love to have your financial support. You can go to microbe.tv/contribute. You know, here at Microbe TV, all the other podcasts, and there are many besides TWiV, we all give you science facts. We don't give you misinformation. So you want to know what's going on, you come here. And we don't do ads, so we need your support, but we are a nonprofit. So if you are in the U.S., you can federally tax deduct your donations, even if it's $1, although you have to donate a lot to get past the minimum deduction, right? That standard deduction.
Yes.
It's a lot. You need to give a lot of money.
If you're itemizing a $1 deduction, you're doing it wrong.
Forget it. You can itemize a lot, and you will still not exceed that standard deduction, which I think is $32,000 this year. So you need to exceed that. I couldn't exceed it.
Well, no, you don't have to exceed that just in donations. The standard deduction is like all the stuff that you get to deduct.
You could put real estate taxes.
Yeah,
on
your
that,
interest on your mortgage or whatever other.
Yes,
there's a bunch of stuff you can deduct.
But
charitable contributions can push you over to.
Yeah, but if you are up there, you can give to TWiV. Well, we should give to TWiV anyway because it's a good cause, whether or not you deduct it or not.
I give to various causes, even though I generally end up taking the standard deduction.
You know, many people give $100 or $200 and they ask for a receipt because they want to deduct it. So they must be itemizing and doing well. So that's great.
Or they think they might be itemizing.
Yeah.
Every year, my husband asks me for a receipt from Microbe TV, and I say, Vincent doesn't send those. It'll be fine. But if I ask for one, I could get one.
Yeah, if you ask for one, you will get one for sure. And you know, a handful of people ask for one. Most people do not. It's just for your information, it's not easy doing the accounting for a nonprofit.
No.
I mean, we have a full-time person here, Valerie, who she's here every day, and all she does is sit at the computer doing financial stuff. Because you have to report every dollar that comes in and every dollar that goes out. And right now we're wrapping up 2024. It's due May 15th, the return. You don't pay tax, but you still have to file a tax return. And it's very detailed. So actually I have her, and I have an accountant who will pay hourly to do it as well. A regular, account, not a CPA, but a regular accountant. And then if you exceed a certain amount of income every year, you have to have a CPA take a look at it and make sure it's all right.
Mm.
And CPAs are very expensive. If I had known this, I don't know what I would have done.
Well, then it's best you didn't.
Yeah. So these are
Because the mechanics of running this are, all I want to do is talk about science. So whenever you give to a nonprofit, just remember there are lots of costs behind the scenes which are not their fault.
Yep.
It's just the way it is. Let's see. We have the last announcement of ASV. This will be the last time you will hear this because May 9th is too late. So ASV 2025 will be in Montreal at the Palais des Congrès de Montréal, July 14th to 17th, asv.org/ASV2025, where you can find the website. That is the website. You can find the program. And there are main sessions, and then there's a satellite meeting the day before, which has a couple of very cool individual meetings. The early bird registration is May 12th, and after that the rates go up, so you should register before that. ASV Cares and Kitty Corp deadline is May 9th. So these are dependent care benefits for ASV members. So that's why this is the last time I'm going to say this. I may say there's an ASV meeting, but no more Kitty Corp or ASV Cares. Jolene,
Yes.
you want to talk about measles? Paul has measles.
Yeah. So, we're going to talk about measles today, and it's something we'll probably be talking about. We have been talking about for months, and there is one aspect, and that's explaining to the adults. But for the adults who want to talk about measles with kids, there's a really great book written by a scientist and illustrated and put together by others as well, called Paul Has Measles. And it's a really nice book for kids to learn what is measles and what it's like to have it and why they don't want to have it and how they can get vaccinated and help protect each other and still go to school. And this book is available in English as a PDF download, and it's also available in 15 other languages. You can find those on Vincent's Virology blog. It's even been read and recorded online. It's available on YouTube in English and Spanish and many other languages as well. So we'll leave the links for those in the show notes for those who would like to have another resource for talking about measles with people who we hope to not have it.
Yes.
So Susanna Lopez is the mastermind behind these series.
Right.
A couple of years ago, she won the L'Oreal Prize, which is a prestigious prize for scientists. And L'Oreal is like a makeup company, right? So
Yeah.
they support women. And she gave most of her earnings towards doing this book. She had it professionally done, So that's really nice. And so she asked me to correct the English. So if you find anything wrong, it's my fault. And I posted links on the virology blog as well. So it's really cool.
There were also follow-up books to
Yeah.
this, Paul and Mosquitoes, I believe.
There's mosquitoes and there's coronavirus also. Follow-ups, yeah. Okay, we have a couple of news stories. Who put up the gold standard story? Was that?
I did. I put that there.
Tell us about it.
So there was an announcement from the NIH yesterday, May 1st, that the Department of Health and Human Services and the NIH is launching a next generation universal vaccine platform they're calling Generation Gold Standard. The idea is that they are going to inactivate whole viruses, particularly focusing on influenza and coronaviruses and other potential pandemic viruses, and that this is going to be better and safer than the current strategies. And notably, it migrates us away from mRNA technologies. So this was just announced yesterday, and it is coupled with the announcement that all vaccines going forward are going to be, if at all possible, placebo control trial tested. And so they sound really good on the surface that you would have a universal flu vaccine and that it would be tested and safe. I mean, all of our flu vaccines are safe. They are tested. They're not all placebo-controlled trialed because that would be expensive and unnecessary based on the history we have with using them. And the idea of the universal flu vaccine is definitely something of interest, but we haven't seen success doing that with using whole inactivated virus in the past.
And you can't just wave a legislative wand or a regulatory wand and say, this will be a universal flu vaccine. It doesn't work like that.
This is Bhattacharya said this is a paradigm shift. This is BS. It is not a paradigm shift. This is the old way of inactivating flu vaccines, which is not very good. Still, we need modern approaches, and they're just going to go back to that. They're going to take different flu strains, including H5, SARS-CoV-1 and 2, MERS, and they're going to inactivate them. And I have to remind everyone that a lot of the SARS-CoV-2 vaccines that were inactivated didn't work very well.
Yeah.
So in China,
Sinovac is a good example.
Yeah, and then they had to follow up with an mRNA vaccine. So this is ridiculous. I saw this as well.
And the placebo-controlled thing, I mean, speaking of flu vaccines, these are vaccines, the ones that are most used, are built on platforms that have been used for decades. And what's done each year is the same procedure with a different strain of flu. We know this works from, again, decades of experience. And so, yeah, you do a safety test in a couple of dozen people, and then it's good to go. And that's really all you need for a vaccine platform that's that well established.
So they're basically going to mix all these viruses together, right? Give you one shot.
Sounds like that's the idea.
They have two candidates. They provide broad spectrum protection in theory, but you have to test them. And how are you going to test SARS-1 and MERS? There's hardly any cases of MERS, and there's none of SARS-1. Did you think of that, folks? I have to tell you something. Well, maybe I shouldn't. No, I'm not going to tell you. This is just very, very
This is
weird.
bad.
It's not good. You know, we have universal flu vaccine approaches that have been worked on for years that involve very clever approaches. Not this. This is not a universal flu vaccine. It's going to hit a couple of whatever strains you put in, it's going to hit. Anyway, I don't mean to criticize your news, Jolene. I just wanted to.
Well, I think it's the thing is when you read the news release, it's not clear why it's not an innovative or likely to work announcement. And so I thought it would be good to talk about it. That way we can add a little bit more information and context to the announcement.
I just don't know. Yeah, I mean,
because if you leave out the history of this field, then it's very easy to say,
that's
we're doing something new. We've invented the wheel. Yeah.
Now, help me understand this. RFK doesn't believe in vaccines, right? RFK Jr. He says they don't work. So this is NIH. He's the head of HHS, of which NIH is a part. So what is this about? Is he lying when he says vaccines don't work? I don't get it. Or is this just a press release and they're never going to do it?
This is sabotage. This is a way to sabotage vaccines without saying that that's what you're doing.
Oh, these are not going to work.
To mandate an ineffective platform and to mandate cumbersome, expensive, placebo-controlled trials for things that have already been proven.
No, it's an interesting idea.
Yeah.
It's a redirection of resources that then might be followed by a removal of resources.
Yes.
That's what it looks like.
Yeah, I mean, especially if they do this inactivated vaccine and then they manage to do a controlled trial and it turns out it doesn't work very well, which we could have told you, and then they could say, oh, well, we're just going to pull funding from that.
So, this is part of this announcement is Jeff Taubenberger, who's now the head of NIAID, right? It's Tony Fauci's old job. And he's a flu researcher. I interviewed him down at NIH a couple of years ago about his lab participated in the recovery of 1918 influenza virus, right? And I thought he was okay. So the only thing I can think of is he wants to keep that job, so he's just going to go along with this.
So far, I mean, he got put into that position because the what's her name? Who took over from Fauci? I just blanked on it. She took over after Tony Fauci
Gene
left.
Marazzo, right?
Marazzo, that's it. And then she left because, see
Yeah.
above, all the nonsense is going on. And so they elevated, the next in line is somebody who'd been there for a long time, who's now in charge.
Yeah.
And he's probably figuring, well, if I leave, then they're going to put somebody else here and we'll just keep, all leaving and there'll be nothing. And maybe it's better for somebody to be here.
Also, I think part of the thing here is that they're making people think that we're still working on vaccines.
Yeah.
Right. We're not abandoning them completely. Although we're just using technology that's going to fail, as you said, Alan. So that's a good point.
I do think that the people who are still working inside of these federal agencies are working under conditions and circumstances that are not great. And so
no.
I actually am grateful to the ones who are still there and who are working as much as they can to make things continue to run. Because if they all left, we'd be in really big trouble.
Yeah.
Did you also put the updates, measles updates in?
I did.
Okay.
Every Tuesday and Friday, I'm in the habit of checking the update released by the Texas Department of State Health Services. So there have been now 683 confirmed cases in Texas. And that's a 20% increase since April 29th, which was the Tuesday update compared to today. And the CDC also reports total cases at 935 for the 2025 year, of which 96% of the individuals who have a confirmed case were unvaccinated or of unknown status. And 13% of those have been hospitalized, including 23% under the age of 5. Still only 3 deaths, but death isn't the only terrible outcome from a measles infection. So I thought that would be apropos to what we're talking about today.
So, Jolene, if you go to the Texas site, there's a map, outbreak cases by county.
There is.
So where are you on that map, roughly?
Well, on my screen, you actually have to scroll over to the right to see it. We're closer to the East Texas border.
Mm-hmm.
How can I describe it to you? Brazos County looks kind of like the state of New Hampshire or Vermont.
I see, yeah.
Sideways.
Yeah. I see. And that's what you're around there?
Yeah, we're in Brazos County.
So you're right on the border, more or less, right?
No. We're actually 3 hours from the Louisiana border. By car.
So the only close county that had a case is north. Those 2 counties north, right?
Yeah.
And close being a
exactly
term,
close being
Texas.
4 or 5 hours away.
Yeah. For me, that New Hampshire county looks like it's just miles away. But yeah, it's a big state.
I mean, you might be looking at another one. If you hover over them, then the county name will appear.
Yeah.
Let's see. Newton. I'm looking at Newton.
Okay.
What's the name of yours?
Brazos.
Oh, I'm in the wrong
It's
one.
more towards the center, not exactly in the center.
Brazos. There it is. Got it. Yeah, it's pretty far. Yeah. The one I was looking at is Jasper County.
Ah, yes.
Which is right near the border.
Oh, yeah, that is right on the border.
Or Newton. They both look like New Hampshire-ish. Okay.
Yeah. So for the most part, the outbreak has remained contained in West Texas, probably because West Texas is a pretty rural area of the state, actually. So there's not as much cross-pollination between that area of the state and the metro centers of Dallas, Houston, Austin, and San Antonio.
People are not commuting from West Texas into Austin.
Correct. You see more in El Paso, which is the bigger city center. That's that leftmost tip there.
Okay. Thank you. All right, I have one news item. This is an article in the Telegraph, which I just found it because it's open. You could read it as opposed to the other places. Top secret U.S. research lab studying SARS-CoV-2 is shut amid safety concerns. So this is Fort Detrick, the integrated research facility, which is a high containment facility. It includes a BSL-4 laboratory.
And Fort Detrick, by the way, is not exactly a top secret facility.
No, it's not.
This is not Area 51. Everybody knows where it is.
Yeah, in fact, Rich and I went a couple of years ago to do a TWiV with a few of the virologists there. And what's interesting about Fort Detrick is that it's got this Army, Navy research base consisting of multiple buildings. And then part of the National Cancer Institute is there as well, which has just civilians, right? Nothing to do with the military. It just happens to be on the same piece of land. You have to go through the gate. Anyway, they were told by email to stop all work by 5 p.m. on April 29th. The director, Connie Schmoljohn, was placed on administrative leave. The doors are locked and you can't go in. And they worked on Ebola, SARS-CoV-2, Lassa fever virus, Marburg virus, Eastern equine encephalitis virus. So this is not good because I had heard that the virus stocks were destroyed. That's not reported in this article. But they said here that the freezers have been padlocked shut. Well, they should be. If they're in a BSL-4, they should be padlocked in the first place. So that's nothing new. It has 168 employees. I think they had lots of animals there. I don't know what happened to them. Again, my other source said they were all sacrificed, but here it doesn't say anything about that. But this is where I don't know if anyone remembers Jens Kuhn.
Yeah.
He's been on TWiV a couple of times, but he works here. He works as an independent contractor, but he can't go in if the place is shut down. So, and I have a feeling his government email has been shut off. So I just sent him an email saying, "Here's my cell phone."
Well, and they're in this article, they quote Bradley Moss, communications director for NIH, who said this is a safety stand down. It follows identification documentation of personnel issues involving contract staff that compromise the facility's safety culture, which is a kind of oblique way of saying we don't like the look of some of the people who work there.
Alan, I don't believe a word of it.
No.
I just don't. It's an excuse because they're all liars.
I think this is just more propaganda. But then again, that's kind of my baseline assumption about anything this administration does now.
I agree. Now, there are a couple of BSL-4s throughout the U.S. So there was another NIAID one in Hamilton, Montana.
Yep.
So the thing is, the research at these facilities is what keeps us leaders and ready to respond when these kinds of, let's say for the national defense, we have an outbreak of a really bad virus, whether it's imported or something else happens. We're going to shut down all the facilities that can do work on this kind of disease caused by, really, there are some diseases that are very bad that you don't want studied everywhere. You need to have appropriate safety protocols and infrastructure. And if we remove all of that and all of the people who worked on that, then even if we have the infrastructure, if we can't staff it, then we're going to be vulnerable to biological hazards, including possible bioweapons in the future.
I read in another article that we don't have Ebola here, we don't have Lassa here, we don't have MERS, so we don't need to work on it. That's just an excuse, by the way.
But it comes here.
It can, yes.
From other places.
Yeah, of course, it could come in big time. It's just crazy. There's a quote from Professor Stuart Neal, head of Department of Infectious Diseases at King's College London. These aren't on-off switches that you restore with a flick of the finger if someone on high changes their minds. This is willful vandalism of research and treatment programs that have taken years to establish, things that you could legitimately say made America great.
Yes. And I've mentioned this several times before in this conversation about they fired this number of thousands of people and so forth. You can't just turn that back on. You can't just bring those people back.
Yeah. Really unfortunate. All of this. Write your senators and representatives. Every week, write them. Go to the demonstrations. They make a difference. When these people see all these people in the streets, they'll say, oh, these are my constituents. They're not going to vote for me if they don't like this. So do it. Do it. Do it.
Yeah.
Okay. Onwards to science. I have a snippet for you. I didn't open. Okay. So, Alan, I think you could read that the key points.
Oh, yeah.
That's
so
a beautiful snippet.
So the summary they wrote for us. It's an odd thing with JAMA. They've got this paper. You can read it online. So in a sense, it's open access, but you cannot download the PDF without signing in.
Yeah.
I found
So
that out
read
at
it
home.
online. The paper is modeling reemergence of vaccine-eliminated infectious diseases under declining vaccination in the U.S. Matthew Kang, Kate Bubar, and Yvonne Maldonado. Oh, there's an et al. On that. Peter Hotez and Nathan Lowe. And they are from Stanford, Baylor, Rice University, and Texas A&M. Okay, so they have this key points at the top, which is very good. The question they're looking at is how will declining childhood vaccination rates affect risk of outbreaks and reemergence of previously eliminated infectious diseases in the U.S., which is certainly a topical question. They found at current state-level vaccination rates, measles could become endemic again. Increasing vaccination coverage would prevent that. Under a 50% decline in childhood vaccination in the U.S., which they modeled, the simulation predicted 51.2 million measles cases over a 25-year period, 9.9 million rubella cases, 4.3 million poliomyelitis cases, 197 diphtheria, 10.3 million hospitalizations, and 159,200 deaths. Bottom line is childhood vaccination at a high coverage level is needed to prevent resurgence of vaccine-preventable infections and all of their sequelae, which is a not surprising message, but it's good that they actually
it's good to see hard numbers, and you're going to see more of them. This is done just at the right time, unfortunately.
So this is really answering the question, how bad could it be?
Yes, exactly right. So a little background. In the U.S., we have eliminated many childhood infectious diseases through vaccination. And we continue to vaccinate because there are always cases which are imported from elsewhere. So the U.S. remains, because people come here all the time, although maybe they don't come here anymore, I don't know. The U.S. remains vulnerable to reintroduction and reemergence. Okay, so we have to continue vaccinating. So you've eliminated measles, but there's a ton of measles in the rest of the world. You can't stop vaccinating because it's regularly brought in by some non-immunized person who then gives it to someone else.
And also, right at this very moment, we have not eliminated measles.
Yeah, we have.
We've got it circulating.
It was eliminated in 2000. Now it's back. It's been back for a number of years. Yeah. And the problem is, of course, the vaccination rates in the U.S. have been declining, and there are actually debates ongoing whether to reduce or eliminate the childhood vaccine schedule, which have all increased since COVID pandemic because people got mad that they were forced to be vaccinated. Are you mad that you have to wear seatbelts also, or that you have to go 55 miles an hour?
By the way,
Yes,
I'm not
they
aware
are.
of anybody being grabbed on the street and given a shot.
Right. Forced is
Forced is a strong word.
Well, you
could
can't
be
go to
forced-
work unless you were vaccinated.
You
for
could be
example.
forced to go to jail if you killed somebody.
Yeah.
You weren't forced to get vaccinated. But a lot of places required vaccination because it was necessary for public safety. And so, yes, your life became more complicated if you continue to refuse vaccination, kind of like it becomes more complicated if you refuse electricity.
Yeah. But actually, it's what's interesting is that there was a measles outbreak in Philadelphia. I forgot, in the 90s maybe or 80s. And it was related to a couple of churches telling their congregations not to get vaccinated. And so the mayor forced immunization. He forced it. He
They
said,
actually went to people's homes.
he went to people's homes
Mhm.
and they forced it. And, of course, the churches sued and some legal organization that normally takes care of these things. Maybe the ACLU, right? They said, we're not touching that. We're not touching it.
This is not a civil liberties issue. Sorry, guys.
But that's the only example I know of. Yes. But it's to do things, you had to be vaccinated for a while.
And, there are many, many other things that we regulate or ban or restrict or force in order to interact publicly, because if you do certain things or fail to do certain things, you endanger others. That's just living in a society.
A society, exactly. We live in a society, and part of the social norms are to let other people live without endangering them. Like we tell people not to drive drunk because there's a real risk that you're going to hurt yourself and others. If you're only going to hurt yourself, there's less of a problem, but we do have lots of regulations on not taking illegal drugs, and most of those only hurt yourself, but still, it's not good for you or society as a whole.
And the argument with a lot of those drugs is that they create other hazards as a result of the supply chain.
Yeah. Okay, so this declining vaccination rate is due to a lot of things: misinformation, distrust, other things, and so it's associated with an increase in the number of outbreaks of vaccine-preventable diseases, like the outbreaks in Texas of measles and multiple other U.S. states that have been happening. And so there's a lot of debate, as they say, on vaccination. So there is some potential that vaccination rates will decline substantially over the coming years. And so this is a modeling study to answer the question, how bad could it get? And they look at either decreasing or stopping routine childhood vaccination for measles, rubella, diphtheria, and polio. All right. Now, let me tell you about the model, because you need to know how they put this together. So those viruses and bacteria, they modeled the number of cases under different scenarios of declining vaccination, and they simulate importation from another country and then transmission across the states. Each individual is assigned to an age group and a state of residence, along with pathogen immunity, and then the model is built on this idea that you are susceptible to exposed, you get, and then you're infected, and then you recover. Right, the typical SIR epidemiological model, and each state was modeled independently. Right, they didn't consider viruses moving across states, which of course would happen normally, but I guess it made it too complicated. And so you can imagine that the numbers would be even higher if viruses are going across states.
This is when building a model. I think generally the approach is, if you've got something that's going to be too complicated to model, then you try to simplify it in a way that would skew your results in a predicted manner. And this would skew the results toward a less bad outcome, modeling the states individually.
Now, polio had a slightly different model, because the inactivated poliovirus vaccine, which we've used exclusively in the U.S. since 2000, protects against paralytic disease. It does not protect against infection. And also for diphtheria, the vaccines don't block infection or colonization, but they do reduce clinical severity. Now, in both cases, they say transmission also, but I'm not sure how. It may reduce it, but it doesn't block it completely. So there are a couple of key things that they use, and one of them was basic reproduction number, the R0, which is basically the number of, if you have an infected person, R0 is the number of people that person could infect on average, assuming that person is in the vicinity of the other people, right? If you're sitting in your home, the R0 is meaningless if you never contact any other people. So R0 was really an important component of this. The model also accounts for age-specific social mixing, which means, I guess, if you're old, you stay home, you don't do anything, right?
Well, I think they might have modeled like people mixing within their age group more than
Yes.
except families. Obviously, you've got across.
Yeah. They also considered you're protected by maternal immunity given through the placenta for 6 months. And the model includes infection-related complications for each pathogen. And they used state-specific birth rates and state-specific and age-specific death rates. Because remember, this is a 25-year period, so population is going to change. So the risk of importation, that's an important part of this model, was based on historical estimates for what we know has happened in the past. And they say these happen most frequently when an under-immunized traveler comes to the U.S. from an endemic country, and then that traveler could come from the U.S., go to an endemic country and come back, or it could be a visitor and introduce the virus or the bacteria. And so they model this importation risk. And again, it's on a state-by-state basis in the total population of the state. So more populated states have a higher risk of, because of the model, the way it's set up, more populated states have a higher risk of importation, which would mean like California and Texas,
Mm-hmm. Yes.
which is having a measles outbreak, probably was
Yep.
imported, right? So go live in a small state, folks.
Well, a less populated state.
Less populated, like
I mean, Rhode Island is a
Massachusetts.
small state, but it's got very high risk
Yeah, I
of
meant
importation,
less
right?
populated. What's the
Well,
population of
West-
Massachusetts?
Oh, that's a good question. It's
Well,
pretty.
you look that up. West Texas, by the way, is less populated than the rest of Texas.
Yeah.
So
But they have an additional factor, which is the low immunization rates. Right?
all states have additional factors.
Yep.
Yep. And for some reason, my browser is taking forever to load.
That's all right.
Vincent, are you going to go over the stuff in the table?
No, I wasn't going to go over that.
I think it's really interesting.
Go ahead. You can.
So the inputs that they used in the model for each of the 4 diseases here, they had basic reproduction number, and you hear about that a lot for measles being the highest out there. And usually you hear the number 12 to 18. And in this model, they use the lower end of the number. They use 12, not 18. For rubella and poliomyelitis, it's 4. And for diphtheria, it's 2.5. And then they have latent period, duration of infectiousness. So for measles, it's 8 days, right? 4 days before and 4 days after the rash. Rubella, it's 14 days. And for diphtheria, it's 14 days. But for poliomyelitis, it's 21 days. And then they show the importation rate, vaccine efficacy. Just a reminder, right? Measles vaccine efficacy is 97%. Rubella is 97%. Poliomyelitis is 90 to 100%. And diphtheria is also 90 to 100%. And then they also show the risk of infection-related complications. And I think this is relevant to talk about because we're talking about very large numbers. Here in the US, there's huge numbers of people. So even if you have a low case number, you rack up the total number of cases and make these risks higher. So for measles, post-measles neurological sequela is 0.1%, hospitalization 20%, and death 0.3%. Then for rubella, congenital rubella syndrome is 65% of pregnancies that have an infection between 0 to 16 weeks. And then death is 30% of cases. For poliomyelitis, paralytic poliomyelitis is 0.5% of unvaccinated cases. And then if you are actually sick, you'll be 100% of the time hospitalized. 10% of those paralytic polio cases die. And for diphtheria, 100% of unvaccinated cases are hospitalized and 10% of them die.
So I have one issue with this, the polio,
Mm-hmm.
which is the importation rate, which is 0.2. For polio, it's not just dependent on importation. We have poliovirus in wastewater. So that means people are infected with poliovirus and shedding it.
Right.
And they are not developing polio because the immunization rate is so high in the U.S. So if the immunization rate drops, you will get cases of polio from within the U.S., not imported. It will be higher than 0.2%. They don't take that into consideration because we don't know how extensive
Exactly.
the poliovirus in wastewater is in the U.S. because the CDC won't test it. The only place
I
they
saw
tested
that. I was like, well, they're only looking at importation. We know we've got
Yeah.
it, but we don't know how much we've got.
Yeah.
So
they looked in the New York area after the case upstate in
Yep.
2022, and they found it in wastewater, and they decided not to look elsewhere. And I emailed someone at CDC, and I said, why not? And they said, that we don't understand what the relevance of that would be. And I said, seriously, I have to teach you basic poliovirus biology? The relevance. Anyway, that's the only weak thing I can see here.
but it's not
Okay.
it's not weak. It's just a limitation of the data.
Well, yes, I think you would have more cases of polio if you
Yeah.
knew how. If you got an
And
idea
again,
of what,
it's a model simplification or assumption or a lack that skews this prediction in
Yes.
the
To
less
lower.
bad way.
Mhm.
So if anything, this is like, what's the worst that could happen?
Yeah.
Best case scenario.
well, I mean, as you'll see, the number of cases of polio is far more than we've ever had before in history.
Yeah.
So it's because population is
Yeah.
much larger now. All right. Couple of other things they use age-specific profiles of population immunity, which they get from national immunization surveys. That's reported in the public literature and also in different states for people 24 years of or younger or 25 years and older. And they also use the risks of infection-related complications based on the literature. So they simulated models under different scenarios of childhood vaccination, as you will see. Now, the primary outcomes were estimated infections with measles, rubella, polio, and diphtheria, and the secondary outcomes were estimated infection-related complications. So, as Jolene mentioned from the table, post-measles neurological sequelae, paralytic polio, congenital rubella, hospitalization, and death. And they detail these furthermore. They note one thing here that's very interesting. I'll go over the measles. For measles, the neurological sequelae include primary measles encephalitis with persisting complications, acute disseminated encephalomyelitis, inclusion body encephalitis, and subacute sclerosing. Panencephalitis. They say hospitalization for measles is often due to complications of pneumonia or dehydration, and more rarely encephalitis. And all clinical cases of polio and diphtheria are recommended for hospitalization. And they did 2,000 simulation runs. You just tell the program to run over and over.
You just keep hitting the button. Yeah.
Hit the button because there's a lot of variation in these estimates.
Actually, you probably don't keep hitting the button. You just have a for loop.
Tell it, yeah.
You just
Yes.
tell it to do it over. Okay, so that's what the model is. And they actually ran some simulations which they compared to historical data, and they gave similar numbers, right? And so they think that it's working. And just remember, it is a simulation, so you just never know. All right, so in 2024, that's the first year of the simulation, there's moderate state-level variation in childhood vaccine coverage. This is going back from 2004 to 2023. So it's 88 to 96% for measles, mumps, rubella, 78 to 91% for diphtheria. That's DPT, diphtheria, pertussis, and tetanus, and 90 to 97% for inactivated polio vaccine. And over a 20, now here are the results. If you're still with us, over a 25-year period with current state level vaccination rates, their model predicts 851,300 cases of measles, and there's a big confidence range, but I'm not going to give them to you. This is, the mean. 190 cases of rubella, 18 cases of polio, and 8 cases of diphtheria. Current vaccination rates, which are pretty high, right? Overall, they're pretty good. They project 851 cases of post-measles neurological sequelae, 170,200 hospitalizations, and 2,550 deaths from the measles. With an immunization rate, MMR immunization rate, 5% higher over 25 years, there'd be 5,800 cases,
which is
less.
A huge amount less compared to 851,000.
Yeah. And with 10% higher vaccination rate, there would be 2,700 cases of measles.
And this is, the difference here is an enormous human cost to these people, these kids who are getting hospitalized and having lifelong neurological complications and all that. But even if you wanted to be, pure hard-nosed Republican about this and say, well, what's the bottom line? The bottom line is that's a hell of a lot of money. My gosh, that's a huge drain on the economy that you can prevent by boosting immunization rates just a little bit. And the vaccine's cheap.
Compared to the healthcare costs and the loss of work.
Yeah.
Mm-hmm.
Okay, now if vaccination rates decline 10% for MMR, 11 million cases of measles. 11 million over the next 5 years. If routine childhood vaccination declines 25%, they're estimating 26.9 million cases of measles. This is, again, in 25-year period. 790 cases of rubella, 87,600 cases of polio, and 11 cases of diphtheria. And then for complications, 26,900 cases of post-measles neurological sequelae, 100 cases of paralytic polio, 1 case of congenital rubella, 5.4 million hospitalizations, and 80,600 deaths for all of these infections combined. Now, what if they declined by 50%, which may not be unheard of, right? Who knows? 51 million cases of measles, 9.9 million cases of rubella. That's very interesting, the rubella, because it's 87,000 cases with the previous decline of 25%.
There are threshold effects going on here.
Yeah, clearly.
So when you drop a little bit, measles comes back right away because it's so contagious. But then the others, because they're significantly less contagious, you really need to drop below a certain threshold. Yeah. And they're modeling, and this is a really useful feature of this paper, what are those thresholds? When do you get to the point where all of a sudden, polio explodes as opposed to measles?
Yeah. So 9.9 million cases of rubella, 4.3 million cases of polio, and 197 of diphtheria. Diphtheria seems to be resilient, right?
Mm-hmm.
51,000 cases of post-measles neurological sequelae, 5,400 cases of paralytic polio, 10,000 congenital rubella. That's very bad. Babies are born with all kinds of problems, including blindness. 10.3 million hospitalizations and 159,000 deaths. And so they do some other numbers, including complete cessation of childhood vaccination. So the differences here, of course, are the timing. And measles is the first to return because of its infectivity. It was predicted to return to endemicity, which means it's continuously circulating and infecting people in 83% of simulations after 20 years, whereas the other diseases would not return to endemicity. But, the percent immunization can affect that.
Right. That's if nothing changes.
Yes.
If nothing changes from right now, which it's already been on a decline.
Yeah.
So if you have a 25% reduction in vaccination coverage, polio becomes an endemic in 11% of simulations. Diphtheria and rubella do not become endemic. 50% reduction, measles returns in 99.8% of simulations after 5 years to endemicity. And rubella returns to endemicity in 100% of simulations at 18 years. Polio in 55% in 19 years. And diphtheria returns to endemicity in less than 1% of simulations and took more than 2 decades. Although they say it does become endemic, diphtheria, when you have even more larger vaccine decline.
So I want to make my cynical comment, which is thinking about declining birth rates has become a national security concern here in the United States and around the world. And you wonder why people don't want to have kids when the world is not getting safer as a place to raise children if you have this kind of an outlook. When it used to be, as said, that the future looked better for me than for my parents. And many people don't feel that way anymore. And even if the data didn't say that, that's how they feel. And these kinds of predictions don't make you feel like it's a great place to have kids.
Yeah.
Yeah.
Yep, for sure. So those are the numbers.
And by the way, that whole, declining birth rates are a national security threat thing that's a whole other conversation. That's not a claim that should be left unexamined.
All right, so those are the data. A couple of conclusions. So first of all, they picked these diseases because they're of high consequence to public health, right? Highly infectious and bad complications. And so their data say that over the long term, they suggest the U.S. may be on the threshold of losing population immunity to prevent transmission of measles, in the next 20 years or so, and the others following suit. And so they say, these findings underscore the importance of high levels of routine childhood vaccination to prevent reemergence of these eliminated infectious diseases. And so this is a counter to what RFK has said, right? He wants to try and eliminate vaccination, especially childhood vaccination. And this is a direct counter to that. You just cannot do that. Hopefully he wouldn't be in office for more than 4 years, and then all of this could be reversed, although it will take a while.
It'll
right?
take a while.
So there were 3 things that drove the increases here. First, the infectiousness, right? The R naught, population immunity, and the risk of importation. So those parameters make a big difference in the numbers they got. And of course, measles is the most commonly imported. Polio and diphtheria are the rarest, although, as we said before, polio is likely to go up. And they say this study gives you data to guide decisions on the importance of routine childhood vaccination. Okay, so the people who are moving to reduce or eliminate childhood vaccination, look at these models and see what kind of a disaster you're going to cause if you do this. Don't just say because you think it's the right thing to do without reading anything or thinking, which I think most of these legislators do. Because once an infectious disease returns to endemicity, they say re-achieving elimination may be challenging without efforts to address the root cause of declining vaccination. Exactly. So you can imagine, if we get to a point where half of the population is not immunizing, it's going to be hard to convince them to go back. Especially if they say, "Oh, I'm fine. There's nothing. I'm okay. My family's."
I don't know.
okay."
After their first kid dies of measles, maybe they'll come around.
The people in Texas whose kids died say I would do the same thing. I would not vaccinate my child. Right. So I don't know. I would think that would do it, Alan, but who knows the way people think. So I think this is an important paper.
Yes.
And people need to think about the numbers. And, Dr. Cassidy, the physician senator from Louisiana. He should be waving this paper when the debates come up about immunization. He really needs to do that because, after all, he has the dual role. Should worry more about people's health than him getting reelected. Okay. Now for something completely different, Jolene.
And by the way, Massachusetts' population is around 7 million people.
Okay.
So your
In case anybody
entire
was still
state's
wondering.
worth of population would have had these infections
Yes.
multiple times over if
Yes.
vaccination rates decline. Yes.
Okay, Jolene is going to do something that's very different.
Yes. Okay. So we're going to talk about a paper called Bacterial Outer Membrane Vesicles Bound to Bacteriophages Modulate Neutrophil Responses to Bacterial Infection. This is from 2023, and it is published in the Frontiers in
Cellular and
Frontiers?
Infection Microbiology.
Thank you. The authors, there's co-first authors Nina Penitzdorfer and Medea Popescu, and this comes from the lab of Paul Boyke at Stanford, but there were also affiliations at the Paris Science et Lettres University in Paris, France. And this is a paper about how phage infection of bacteria influences bacterial infection outcomes and why. And the big question is, how does the presence of a particular kind of phage change the response of the eukaryote or the human host to a bacterial infection? So there's like two hosts here, the bacterial host for the phage and the human host for the bacteria, although they also do this in mice. And the observation is that there are these certain kinds of phage called filamentous phage that are lysogenic. They integrate their genomes into the chromosome of the host. And these are filamentous phage, so they're very different from the phage that I usually talk about. They are just like rod-shaped.
They don't look like that.
They don't have tails. They just look like a tail. They have single-stranded DNA genomes. Yes, like that. No tail, no tail fibers, just a long rod. And then at the end of the rods are these minor proteins that are responsible for interaction with the host. And they have very different properties compared to the tailed phages. They have an overall negative charge, and they are what we call extruded from the host cell surface. So there's no envelope. It's a protein that coats the DNA complex. And the DNA is packaged in a very strange manner. It's what we call P-form DNA, actually. So the bases are kind of flipped out. It's very loosely packed. And the extruded particles come out from the surface of the cell. These are gram-negative bacteria, so the outer layer of the outer membrane. But they do not lyse the cell. So the cell does not have to pop for these viruses to get out. And they have, because of this negative charge, they have these ability to aggregate, or not aggregate, associate and form what we call liquid crystals. And those liquid crystals can alter the properties of the environment in which the bacteria is located. So they are shown to change the diffusion of antibiotics into biofilms formed by Pseudomonas. And this is a major problem for infections in humans, infections of wounds, as well as lung infections in patients that have cystic fibrosis, for example. So having this biofilm with this phage that has formed liquid crystals is going to prevent the host immune response from responding the same way that it would, and it prevents antibiotics from working the way that you would hope they would. So there's a lot of interest in understanding how the phage presence as a prophage or as a lysogen is going to change the infection dynamics in the host. And they had previously seen that these phage the presence of these phage would provoke a response to a bacterial infection that was more like a response to a viral infection. And so that was unexpected, and that's really what they were trying to figure out here, was how was the phage causing a response as though it were the infection or with a virus.
And did we say these phage, in this paper, we're talking about a phage of Pseudomonas?
I did mention Pseudomonas.
Okay.
I didn't mention the name of the phage. This particular phage is called PF4. Okay, so the overall conclusion is that the presence of these phage seems to be redirecting the immune response away from bacterial signals that the immune response usually responds to, and it is actually responding to bacterial debris or bacterial molecules that are stuck to the phage, that are not necessarily part of the phage structure, but that are there. And they are delivering, the phage particles are delivering those molecules in a different way to the eukaryotic cell than it otherwise would be exposed. Okay, so what else can I tell you about the background? When these filamentous phage are extruded from the host cell surface, they often have lipopolysaccharide, which is a bacterial lipid, on the surface. And lipopolysaccharide is also known as endotoxin. That is something that is a potent activator of the immune response, and it causes a lot of inflammation. So you would think that this LPS bacterial molecule would be driving the responses of the human host to a Pseudomonas infection. But
And I just want to chime in one more thing about Pseudomonas.
yeah.
A reason why this is such an important bacterium is that it causes these opportunistic infections, especially in people who are immune compromised or have cystic fibrosis. It's really bad in them. And it can be very, very difficult to treat, partly because it doesn't seem to stimulate the right kind of immune response.
Mm-hmm. That's right. So
And one thing I didn't appreciate before is that, when you make phages for phage therapy, you have to get rid of all this endotoxin.
yes,
So they
that's
purify
correct.
them extensively. Yeah.
So when phage are being purified, the idea is to get rid of all the bacterial debris. The primary problem is endotoxin, or the LPS molecule, because that's a major activator of the immune system. But there are also other bacterial molecules that can activate the immune system, and so we extensively purify them to get rid of that. Nonetheless, once you put the phage in and it's pure and it infects a cell, then it comes out of the cell and releases all of those potential activators. So the question of how that interacts with the immune system is of great interest. And here, these filamentous phages are not really the same or under consideration per se for use in phage therapy, but they're present already in the bacteria that are in the host. So we want to know how that affects the overall pathology. So then the specific response that they're looking at in this paper is the chemotaxis of neutrophils. So you have an infection, usually the host cell will detect those bacterial molecules, then cytokines are secreted. These are signaling molecules that recruit other immune cells that can help to clear the infection, get rid of the bacteria. But when the PF4 phage is infecting Pseudomonas, then there seems to be less recruitment of these immune cells. And in particular here, we're talking about neutrophils, which have, they're similar to macrophages in the sense that they can phagocytose other cells, and they also have granulocytes that they can use to degrade the offending invader. The one other piece here is that the bacterial debris that I'm talking about and the LPS, what they found is that it seems to be associated with these outer membrane vesicles. So Pseudomonas, being a gram-negative bacterium, has two layers, two lipid bilayers called the inner and the outer membrane. The outer membrane is where the LPS is, and under different kinds of conditions, stress conditions in particular, such as when this PF4 phage is extruding and having a continuous chronic infection, there are release of outer membrane vesicles. And those outer membrane vesicles include LPS and other lipids, but they also seem to package other things, proteins and RNAs. So the presence of these RNAs in these outer membrane vesicles is the particular activator that we're going to be looking at in this paper. So they used 2 different strains of Pseudomonas, one that didn't have any PF4 phage in it and one that did. And then they compared those 2 phage by doing a purification, those 2 preparations by doing a purification of the phage. So they took the cells that were infected, they purified the supernatant with the phage in it by kind of standard protocol with polyethylene glycol precipitation. And when they do that, they did it once, and then they can look at those particles. And then they also did it twice to get rid of extra debris. And then they looked at those particles. And they measured how much LPS was associated with the particles. And if they did just a one-time purification, they had a higher level than if they did a 2-time purification. And that was one thing. There was LPS present in the purified particles. Then they also looked at those particles by transmission electron microscope, and they saw, it's a little hard to see in their pictures, but they saw these little small balls at the end of the filamentous phage, and those they are interpreting to be outer membrane vesicles. So they checked that 2 ways. They used immunogold electron microscopy, where they take an antibody that can bind to the proteins at the tips of the filamentous phage, and that has a gold particle attached to it. And that gold particle makes a nice little black dot in the electron micrograph. And so they can see that when they do immunogold against proteins that are known to be in outer membrane vesicles, they get an accumulation of gold dots at the end of the phage in their images.
This idea that there are vesicles on the phage is new to me. I didn't know that. It's
Me
cool.
too. Yeah.
So I knew about this in the context of tailed phages, because that's where I pay the most attention, because the debris that the phage are released into is immediately a danger of inactivating the phage. On the part of the phage, it's ideal to get away from the debris so that you don't accidentally attach to the protein that you usually use to get into the cell on the cell that's dead. So you can see lipid vesicles attached to the ends of tailed phages. I hadn't seen it for these filamentous phage, but it looks like they are here.
Yeah.
And they used another method that I'm not so familiar with. This is called coherent anti-Stokes Raman scattering microscopy. And I tried looking it up, and the gist that I understand is that this is a way to image lipids, because the lipids will scatter the light in a different way than happens with other molecules. And so they took this purified phage prep, they put it into a confocal microscope, and they looked at where they saw the signal by this CARS microscopy. And they saw that it was located at the ends of the filaments of the phage. Right. Okay. So they have phage preps with what look like lipids probably on the end, seem to be associated with outer membrane vesicle proteins. So next what they did was they exposed mouse macrophage cells from 2 different sources to these phage. And they were both exposed to phage in the presence of outer membrane vesicles, and then no phage, but just outer membrane vesicles, and looked at where did the signal inside the cell end up. They have very nice stained cells here. They're looking at the presence of a toll-like receptor, TLR3, in the cell, and that is stained here in red. And then they have the outer membrane vesicles in green. I think I have that right. Yes, outer membrane vesicles in green. And so they show that when you have the outer membrane vesicles alone, these seem to be getting into the cell, and they are not co-localized with the TLR3. And TLR3 recognizes RNA. But when they have the phage that has been 2 times purified, plus outer membrane vesicles, then they see some co-localization that they didn't see before. And they check, So outer membrane vesicles by themselves would be getting into the cell by a different mechanism than outer membrane vesicles that are associated with phage. So they used these inhibitors to see what kind of entry was occurring. Usually I pretty much mostly think of clathrin-mediated endocytosis. And it turns out that there are other ways for extracellular vesicles to get into the cell. So there's endocytosis that is dependent on microtubules and actin, and that's based on kind of the size of what gets endocytosed. And there's endocytosis that is not dependent on the clathrin molecule. So they had a set of 4 different inhibitors. These inhibitors were going to, each of them would inhibit a particular kind of entry mechanism. And they checked the effect of each inhibitor on outer membrane vesicle uptake and on outer membrane vesicle uptake in the presence of 2 times purified phage. What they saw is that even though they could try all of them, the main difference between them was when they inhibited with cytochalasin D. So cytochalasin D inhibits actin polymerization, and that is important in lipid raft uptake. So this is not something I was very familiar with. Apparently, these outer membrane vesicles can interact with lipids on the surface of the mouse cell where they're in a particular configuration called a lipid raft. So you have like enrichment of cholesterol and other lipids that associate with it. And then that can induce the uptake of these outer membrane vesicles more so than if it were at another place on the membrane. That was the only one that showed a difference between plus and minus phage. The other ones, there was an inhibition of uptake of outer membrane vesicles with wortmannin, which inhibits PI3K-mediated endocytosis, nicotazole, which inhibits microtubule-mediated endocytosis of large cargo, and then chlorpromazine, which also inhibits clathrin-mediated endocytosis. So all of them had somewhat of a reduction compared to no inhibitor, but only cytochalasin D had a difference between plus and minus phage. So overall, this was suggesting that the presence of the phage changed where outer membrane vesicles ended up when they were added to mouse cells, mouse macrophages from a couple of different sources.
So the phage is kind of targeting these outer membrane vesicles to a particular route.
To a place that it wouldn't have gotten before by a route that it didn't take before. So now they wanted to know what was in these outer membrane vesicles that would be having an effect on the host, and they purified from these vesicles small RNAs. They went through a purification that included getting rid of surface proteins, so proteinase K. They used DNase to get rid of all the DNA, and then they extracted the RNA, and they checked the RNA for how big it was because it is known that small RNAs have a characteristic size. And they saw that they had a large peak for these small RNAs that are typically found in outer membrane vesicles. And they had those small RNAs in outer membrane vesicles that were formed both in the presence and absence of phage from Pseudomonas cells. Then they took these small RNAs and they exposed human macrophages to them. And these human macrophages were mixed with these small RNAs for a day. And then they took the supernatant to see what was the response of the human macrophage. And they were looking in particular for interferons. Here they were looking for interferon beta levels. This is an important cytokine signaling inflammation and can help attract other immune cells to a site of infection. So they used a couple of different ways for testing the levels of interferon beta. They used an assay called an ELISA, which checks for the protein in a dish, and then they use these reporter cells that will give you a colorimetric readout if there's interferon beta in the supernatant. So they saw that RNAs from the outer membrane vesicles had the highest level of interferon beta response compared to even their positive control, and they could reduce that if they degraded the RNA. And then they took the media from those cells that had been exposed, and they added them to cells that reported the activation of TLR3, which is that toll-like receptor that responds to RNA, double-stranded RNA in particular. And they saw that outer membrane vesicles added to the human macrophages and then the supernatant added to these reporter cells had a high level of TLR3 activation. And they also saw that it was even higher when those outer membrane vesicles were mixed with 2 times purified PF4 phage. When they had just 2 times PF4 phage, 2 times purified PF4 phage, there wasn't really that much of an activation of TLR3, which we would expect because they got rid of most of the outer membrane vesicles if they purified twice. So this suggests that the outer membrane vesicles themselves can activate TLR3, but there's even more activation when they're mixed with this phage. So I presume that the outer membrane vesicles will associate with the phage when they're mixed, and that is what changes their localization and ability to be detected by this TLR3.
And then this complex, the outer membrane vesicles and phage, are taken up by endocytosis, and it must be that in the endosome, which is where TLR3 is, the vesicles are dissolved and the RNA comes out to stimulate TLR3.
Right. Yeah. Exactly. So that was the hypothesis, is that now the outer membrane vesicles have gotten into the cell where TLR3 is. So they wanted to know what kinds of RNAs were in there. And they took an interesting approach. They didn't really talk about exactly the identity of the RNAs, but they did sequence them, and they checked their lengths, and they predicted their secondary structures, because TLR3 responds to double-stranded RNA. So they wanted to see these small RNAs that are in these vesicles, what kinds of structures did they possibly have? They predicted the fold and the energy, that the free energy that would be associated with those folds, and then they use that to predict how much was there that could activate TLR3. And they saw that there was a high, that was basically that the distribution didn't change if there were outer membrane vesicle RNAs in the presence and absence of phage, which tracks with the results that they had seen before. They still make outer membrane vesicles in the presence and absence of phage. It seems like it's just a matter of how it gets to where it's going in the cell.
Right. And this phage, this PF4 phage, is a single-stranded DNA phage, but it's delivering this package that delivers a double-stranded RNA, which fires up TLR3.
Exactly. Yeah.
And that RNA is
Sneaky.
coming from the Pseudomonas, from the host.
Sneaky. It's very sneaky.
Yes.
Well, sneaky would be if it was intentional.
I understand.
Are you sure it's not?
Well,
This is Pseudomonas we're talking about.
No, it's not intentional.
No, it's not.
But it's a very interesting way of getting a different response.
Yes.
yeah. You wonder, how does such a thing, presumably this is a continued phenomenon in nature, and is it a selectable trait? That's not clear. That part's definitely not clear.
It should be, right?
Well, the interesting thing they talk about at the end is that the phage particles themselves are overall negative charge, and LPS is also negatively charged. So it's not entirely clear why these vesicles are associating with the particle. There must be something special about these minor proteins on the end of the phage that encourage the association. So, yeah, you could imagine that you could select for a phage that has a lower outer membrane vesicle association.
And from the phage perspective, evolutionarily, first of all, by being lysogenic like this, it's allowing its host to survive while it reproduces and poops itself out of the membrane. And this delivery of outer membrane proteins is a cool trick that also helps the host survive because it potentially has an effect on the immunity against Pseudomonas that might otherwise take it out, right?
Yeah, maybe. Outer membrane vesicles have multiple roles. So the interactions with the human host maybe are less clear so much as the outer membrane vesicles usually move things between bacteria. But
so maybe this
obviously
evolved for a different purpose and just coincidentally happens to interfere with immunity.
It's interesting because Pseudomonas is everywhere. It's everywhere
Yes.
in the environment and in the pathogenesis cases here in causing this pathology. So, yeah, more to be investigated, I think, to really understand what's going on there. So what they did next is they wanted to look at the downstream effect of signaling via TLR3 instead of via the LPS pathway that typically would be turned on upon exposure to these outer membrane vesicles. And they were interested in inflammatory cytokines and type 1 interferon. So they again took their purified phage and they added that to human macrophages in the presence of LPS. So LPS should turn on the cytokine profile and the kinds of genes that will attract other immune cells. The one that they're looking at is called CXCL5. CXCL5 is an important chemotaxis protein for neutrophils. So when these human macrophages were exposed to the phage, either 1 or 2 times purified in the presence of LPS, they saw that CXCL5 was reduced. It was reduced the most in 1 times purified phage, but it was still somewhat reduced in the 2 times purified phage. And they also saw that TNF alpha, which is one of those signaling inflammatory cytokines, was reduced the most in the presence of 1 times purified PF4. Both of these were measured by ELISAs, looking for that specific protein in the supernatant from the exposed cells. So what they next did was, which I think is a really cool assay, I've never done this before, it's called Luminex. And it's essentially a multiplexed ELISA that allows you to profile many, many different kinds of cytokines. And from having investigated this previously, the number of cytokines or proteins that you want to profile determines the cost. So it's, I think here they had something like 18, maybe something like 20. And what they did was they checked for a large range of cytokines that might be relevant to this kind of inflammatory response they were interested in the supernatant. And they saw that compared to LPS alone, the LPS plus outer membrane vesicles had a really high response. And the more you added phage, so 1 times purified phage or 2 times purified phage, it shifted the profile of cytokines. And they did a couple more assays looking at how this signaling was dependent on TLR3. So they checked again in human macrophages, and they used an inhibitor of TLR3 signaling. And when they used an inhibitor of TLR3 signaling, then they no longer saw that decrease in CXCL5, the neutrophil chemotaxis protein, when the phage were mixed with outer membrane vesicles, suggesting that it really was signaling through TLR3. And then they had an interesting question, I thought, which was, is this a PF4 phage specific thing, or do all kinds of phage that can kill the host do it? So they purified phage by the same polyethylene glycol precipitation method. 2 phage, one called OMKO1 and another one called TVIP, and then they checked to see if that resulted in a reduction in CXCL5 relative to the LPS. but they did not see a reduction. So it looks like other phage that you purify by this same method don't necessarily reduce that CXCL5.
That experiment actually had a little bit of a whiff of reviewer 3 to
Yeah.
me. But maybe not.
So now they've shown that these outer membrane vesicles get in a different way and deliver RNA and seem to activate TLR3, and activation of TLR3 seems to be the method by which they get reduction in the production of CXCL5, which I told you is a neutrophil chemotaxis protein. So next what they did was they purified neutrophils from human blood, and they looked at neutrophil migration. So neutrophils are produced in your bone marrow, and they are released into your bloodstream, where they circulate for a little bit, and then they migrate into your tissues. They live for a few days. But that migration throughout the body and where they end up and how likely they are to end up at the site of an infection is because of their response to these kinds of cytokines and chemotaxis proteins. So they have this neat assay where they use trans wells. So there is a well of media, and then there is a separation barrier, and they put the cells on top of the separation barrier. And then underneath, they can put in the solution, which is exposed to the top layer via permeable barrier, the molecules that they want to see if that causes the neutrophils to migrate. And here they used the supernatants from the cells that had been exposed to the LPS or LPS plus 2 times purified phage. And here I noticed they're using LPS instead of outer membrane vesicles. What they see is that in the presence of LPS, they call that 100% migration. When they add 2 times purified phage to that, they don't really see a difference. But if they add 2 times purified phage plus outer membrane vesicles, they see a reduction in migration. So there's less neutrophil migration when the phage is with the outer membrane vesicles. If it's just phage 1 times purified, there's a reduction, but it didn't seem to be statistically significant. And they also use the TLR3 blocker, the antagonist, to check migration in the assay. And they see that in the presence of the agonist, they no longer get that reduction in migration, suggesting that it still requires TLR3. And then the last thing that they did was they checked all of this in an actual mouse model of infection. So here they have an acute Pseudomonas aeruginosa pneumonia model, and these mice are going to be infected with Pseudomonas, and then at a certain time, a day later, they sacrifice the mice and they wash out the lungs to see what is going on in the lungs, both in terms of the Pseudomonas as well as the response of the immune system to that infection, plus and minus the phage. And they check that response both by ELISA to look at the proteins that were produced, as well as by flow cytometry to check the actual cells. So here they'd be looking for neutrophils. And here, the bottom line is that the outcome is consistent with what they saw in vitro. So they see that the presence of the phage with the outer membrane vesicles has the biggest effect on the CXCL5 protein, as well as TNF alpha and others that they checked. And they see that neutrophils come in more when there's no phage, and they come in less when the phage is there, suggesting that the presence of PF4 in the Pseudomonas aeruginosa population is going to alter the signaling that the immune system can do, and therefore it can reduce the recruitment of important immune cells like neutrophils that can help to clear the Pseudomonas infection.
And this inhibition is dependent on OMVs on the phage, right?
Right.
So why is it an inhibitory response, through CXCR5? What do they know? Because normally triggering TLR3 is a positive thing, right? So
So why does TLR3 reduce CXCL5?
Yeah.
I'm not sure.
They don't actually talk about it.
Yeah.
They do say that it's consistent with known pathways of inhibition, but they don't tell you what they are.
Yeah,
Right.
I was a little confused about the immunology, but
I agree because immunology is not my primary area.
Yeah.
And so
None of us.
anytime that I read these things, they'll say like, oh, this protein is associated with this. And most of the time I just kind of have to believe them.
Right.
I can look it up and see that it's consistent with what other people have said. But how do you pick that one compared to other ones?
Yeah.
I usually assume it's because it's the most important.
Yeah.
Or has the greatest effect.
I mean, it's clearly downregulating CXCR5, but I don't understand the mechanism.
Yeah, they definitely don't understand the mechanism yet either.
In fact, multiple, not just CXCR5, but multiple pro-inflammatory cytokines.
Mm-hmm.
And so I'd like to know what the mechanism is.
Yeah.
They don't even discuss it in the discussion.
No.
But again, they're not immunologists, I guess, right?
No, they do. They do. There's so the corresponding author on this paper is the postdoc who was in the lab, and she has since moved to Gilead and is working on HIV. But the lab that this came out of, they definitely do work on immunology, but they do work on many different things. So it's the lab of an MD, and they have lots of studies looking at immunology related to phage and bacterial infections and viral infections, actually.
But the upshot of this is that this appears to be a mechanism that skews the host immune response against the Pseudomonas infection so that it's not as effective, probably.
Right. Mm-hmm.
Maybe some of those small RNAs have a specific effect on signaling, right?
Yeah, I do wonder about the identity of the small RNAs. Are they important? Can it be any small RNA, or does it have to be a specific one? Maybe
Neat
they
paper.
don't all make them.
Yeah, it's very interesting.
And it's open access. And the graphical abstract I thought was quite good.
Mm-hmm.
So right up near the top, people looking at this paper, check out the graphical abstract. It really kind of walks you through what their conclusions are going to be. I do wish that they had had a copy editor go through it.
Yes.
There are quite a few typos.
Well, that's kind of classic for this journal.
So do you think,
I know.
Jolene, they raised this question as well? Is this a general property of filamentous phages?
Yeah, I was going to say that this is something that has really only been observed recently. This is like new stuff within the last decade or less. So we haven't had much of an opportunity to investigate it. But this group and a couple of others are really pursuing this quite hotly. And they have a review that came out in 2020, so now it's pretty old, where they talk about a couple of other examples where we've been looking at this kind of thing. But it's just kind of an open area that there are a lot of questions unanswered. Like, what is the actual signaling pathway? Is it going to be the same for every phage? Is it even the same in the lungs as it is in a wound? That part is not clear, actually. So, yeah, I think it's exciting though. And I think it's really important work because this is a nuance, right? When we talk about phage therapy, we talk about all the ways that we hope that it's going to make the infection go away. But in this case, there's a phage that actually is probably making the infection worse. And there's even been some work looking at doing vaccination against the PF4 phage, and that having potentially better outcomes on a Pseudomonas infection clearance.
Interesting. Cool. Thank you, Jolene.
Yep.
I think we should do 3 emails.
Sure.
This is quite long. Alan, can you take that first one?
Yeah, let me get things adjusted here. Okay, John writes, Doctors TWiV, episode 1211, Anti-Vax Motivations. Way at the end of Paul Offit's Autism's False Prophets, he reveals that Andrew Wakefield's motivation is religious. I had been waiting for that. Maybe that isn't every anti-vaxxer's motivation, and maybe it isn't the motivation of all those they follow, but I think it's true that the motivation of the original anti-vaxxers that the smallpox vaccine begat was religious. And further to the religious bias of current politics, Barry Goldwater anticipated the current mess. His quote is easily found. Mark my word, if and when these preachers get control of the Republican Party, and they're sure trying to do so, it's going to be a terrible damn problem. Frankly, these people frighten me. Politics and governing demand compromise, but these Christians believe they are acting in the name of God, so they can't and won't compromise. I know, I've tried to deal with them. Interesting.
Back when people said things straight, huh?
Yes. So, yeah, Andrew Wakefield's motivation was partly religious, but it was also enormously financial.
Yeah.
He was running a monetary scam.
Absolutely.
And then John continues, episode 1212, Measles versus polio, what was the difference? About 1% severe outcome with each, right? But polio had poster children, and those were iron lungs, braces, and crutches. A potent visual. But we saw the walking wounded among us. We could easily imagine being in that situation. Just being hospitalized is harder to imagine unless you're at the bedside, and with HIPAA, there probably isn't a hospital anywhere that would let anyone even ask for a consent form for taking a picture of a hospitalized measles patient. It's all behind closed doors now. I know you are being jokeful with, quote, kids don't transmit viruses. I don't remember
we're
that,
joking.
But here's a direct example. My neighbor works in a cabinet-making shop and was recently complaining about how the owner lets his young kids with runny noses run freely there. A few days later, when I saw him, he warned me not to come close because he had some respiratory crud. A week or so later, I learned that another neighbor's 3-month-old baby was in the ICU with RSV, which she also had. He's afraid they probably got it from him. Wow. Well...
Kids do transmit viruses.
Kids
and so
do transmit
adults.
viruses. And,
They do.
And if something was circulating to the kids of the cabinet maker, the 3-month-old baby could have caught it from somebody, just in the community
never
who
know.
already had it.
Never know.
Because if one kid in the community has a runny nose, probably they all do. Episode 1213, microbiome providing natural adjuvants, is exactly what I was thinking when Rich said it. And back to Kennedy with his miasma, it's ironic that less than perfect hygiene at an early age is actually somewhat protective with polio, but people swallow his BS that the polio vaccine kills more than the virus because they have no sense of history. Probably fewer than half can correctly correlate paralysis, polio, and FDR. Don't let up. People writing you to avoid politics are living in the past when politicians got actual experts to speak on issues that were outside their expertise. We are sadly no longer in that arena. I post about this on my Facebook stream and just copied Lisa from Minnesota's letter to you. Meanwhile, Greater Braddock was in the direct path of the 70-plus mile per hour winds late Tuesday afternoon that hit the Pittsburgh area and killed power for nearly 250,000 people. This made it hard to send this email earlier. By Thursday morning, only about 43,000 were back with power. I was one of the lucky ones. My power was restored after only about 28 hours, and I had only minor tree damage. Well, thank you, John.
Yes. Yeah, Jolene, take the next one.
So Mike writes, hello, TWiV crew from Mike in New York City, where it's a clear and beautiful 17C62F. I strongly recommend that we look at public health on a global basis as a matter of United States national security. I never came up with the right wording for this argument, but I recently found the following discussion with Atul Gawande, who makes the best case that I've heard or read to date. I encourage everyone to listen to this discussion. And there is a link to the Lawfare Daily, Why Public Health is Critical to National Security. And Atul Gawande is a surgeon and a public health expert. He's the former head of global health at USAID, an agency that the Trump administration has prioritized for dismantling since its first day in office. on the episode that is linked here, the editor, Natalie Orpit, sat down with Gawande to discuss what USAID does, the consequences of destroying it, and why public health is so important to U.S. national security. The episode that is linked here was recorded on March 27th, 2025, a day before the Trump administration announced that it would be dissolved by the end of the fiscal year. Best regards from Mike. And so I took a look at this. I thought, let's see here. If I can get back to it. So there were a couple of interesting things that this interview went through. So the main thrust was that USAID does very important things. And I think that the person who was doing the interviewing, Natalie Orpit, she tried to play the role of someone who takes the stance that infectious disease, some people die, sad story, but tell me how that really matters for national security. So it's true that there is a toll. He says that, 15 million people died in less than 2.5 years during COVID. And that usually is only something we see during a war. So you would think that that might be an argument that actually made a difference, but apparently not. So in the case of a skeptic, his responses were things like, we need weapons against threats to our country, and weapons are not just guns. They include being able to deploy capabilities around the world and in our country to stop disease threats as they emerge. He talks about measles and vaccination. And then they also talked about how the Department of Defense was important in helping to respond to disease outbreaks in other parts of the world, which is really important if you have troops deployed somewhere else and there's a disease that they are going to encounter. You want to know how to prevent your troops from getting sick, because if your troops get sick, then you're not going to do very well in the war. I mean, that was a big problem, right, with the 1918 flu. I'm sure there are other examples. I have this, my history is not that great, but wasn't Alexander the Great, didn't he die from an infectious disease and then his army could not win the war?
It's a long-standing problem that in any war or armed conflict, typically far more of your troops will die of infection than of enemy action.
Right.
And I would add to this that there is a large number of people in the Department of Defense, including in high positions and in the intelligence agencies, who are very keenly aware that public health is a national security issue. And who I'm guessing are kind of tearing their hair out at the moment with what's going on, because not only what you just said, all these issues of the impact of infectious disease on countries and on the troops, but also infectious disease outbreaks are incredibly destabilizing.
Right.
And the more places get destabilized, the worse off we are as a country, because it's these places that are falling apart that cause the threats that are coming to us. So yeah, this is a big deal, and it's something that is only now being disregarded by, the current administration, but the folks who are responsible for national security are well aware of this, I know.
Yes, it is another example of malignant incompetence.
Yes.
Right? The desire, the stated desire to stop having other countries screw the U.S., which is not the case. We're helping them, and not realizing the benefits of us helping them. Because you let this idea that we're being screwed override everything. By the way, Jolene, there are several ideas about what killed Alexander the Great.
Okay.
One of them is a Campylobacter infection.
Mm.
And another is West Nile encephalitis. I have here an article in Emerging Infectious Diseases where they posit that, but it's hard to know.
So whether it was a bacteria,
he had died within 2 weeks, right? So some infectious disease. Yeah.
Yeah. There was one other point that Atul made in this podcast that I thought was interesting, besides the fact that the strength of our economy is because the U.S. productivity increase due to our research and development capabilities, and that we have been positioned as the global leaders in that. And that was that the basic science that comes up with the fundamentals that you send with your troops or keep with your troops who are protecting the homeland, and their personal health is very important. When you're distracted with even health issues at home, then you're less likely to be ready to respond to any concerns that come up.
All right. Zaharula writes, hello, TWiV team. I wanted to thank all of you for your passion about speaking the truth and doing the right thing for science. Thank you also to the CDC epidemiologist who was the editor of EID and who was fired. To this individual and to the Minnesota epidemiologist who wrote in, thank you and your colleagues for all that you have done for public health and for all of us, because a strong public health program benefits every single one of us, not just here in the U.S., but across the globe. I will also thank you going forward for all that you will do, because I have hope that you will be back. What do you mean back? We're not gone.
She's referring to the
Right.
editor of EID. And
Oh,
Mhm.
oh, okay. I assumed it was us. Thank you especially to Dr. Mingarelli for making the point about taking the time to actually speak with people who may have differing views, but who truly don't understand either the science or the disease process. It does take time and energy, but it is truly worth it. And you are all correct that the lay person truly does not care about data points, graphs, or results. They respond to stories. They want to know if this could happen to them or their family. With regards to vaccinations, as clinicians, we can still share the data, but we really need to spell it out in ways that lay people understand it. So I've started telling parents who have vaccinated their children the following. Thank you for vaccinating your child. You are protecting him/her from complications of these diseases, including blindness, deafness, seizures, jaundice, vomiting, diarrhea, dehydration, respiratory distress, stopping to breathe, meningitis, brain infection, sepsis, shock, being on life support, chronic liver disease, infertility, not being able to have kids, liver cancer, cervical cancer, anal cancer, mouth-throat cancer, being paralyzed, and even death. Thank you for caring about your child as much as I do. Many parents seem really surprised when I say this, but this is where the data about the effectiveness of vaccines gets translated so that the layperson understands why vaccinations are so vital. They also respond well to positive praise and positive emotion from their clinician, and then they may go and tell their family and friends about it. They need to understand that we really do care about their child. Talking about the community at large doesn't hit home the same way. They don't care as much about protecting the immunocompromised unknown person or the person who cannot get vaccinated. They care about themselves and their family. In addition, I think for all of us, scientists, epidemiologists, clinicians, we need to look at this through a different lens. For scientists from the other side of the bench and for clinicians from the other end of the stethoscope, because at some point, every single one of us, even RFK Jr. and Donald Trump, is going to be a patient. Trump has been in October 2020 before the vaccines were approved, and he was hospitalized with COVID-19. We all need to advocate from the patient perspective for all of us, whether it's heart disease, cancer, or infectious diseases, given drastic changes in our environment, climate change, and our close contact with other species, and our interconnectedness with each other as humans. Not one of us is immune from having a serious illness or from becoming a patient in need of scientific and medical expertise. We all need to speak up, lay people, parents, scientists, epidemiologists, academicians, and clinicians alike, to advocate for one another, not only for the truth in science, research, public health, and medicine, but also against all the financial cuts in science, research, public health, and healthcare, which will impact every single one of us. Because infections know no boundaries. Climate change is happening. We don't all live in our own little bubbles, and no one is safe until everyone is safe. Thank you again for all the great science communication that all of you do and for speaking the truth. Grateful for Microbe TV and an ongoing supporter. Zaharula is an MD in Eastern Virginia Medical School.
Thank you,
So,
Zaharula.
Mm-hmm.
I think this is important that physicians need to play a big role, right?
Sure.
And many of them are anti-vax.
But not
It's a
all
min-
of them.
It's
didn't
a,
say all
it's
of
a
them.
small
Does
minority.
many
It's a
all? Many is
small
not all. I said
vocal
many of them.
minority.
Many are, and many give out vaccine exemptions when they're not warranted. And those are not physicians who are going to sit down and say this to the patients, and they need to. Now, I understand there are other issues as well besides the physicians, but the physicians are the ones, they're the last connection with the vaccination. Of course, if a patient never gets to a physician like the Mennonites, this is all out the window. However, what we have to avoid is RFK's company, whatever, Save Children, the anti-vax company, going to Texas and telling the people not to get vaccinated. We got to avoid that. I don't understand how they're allowed to go in and do that. Anyway, thanks. Zaharul, that was great. I really appreciate it. We're getting a lot of emails about this issue and I'm glad because I'm happy that even though it's the TWiV people and they're all converted, I'm happy that this is resonating with a lot of people. All right, let's do some picks of the week. Alan, what do you have for us?
I have a book that actually we suspended doing picks of the week during the pandemic, and I started doing them on my blog at the time. This was back in 2020. And I was just moving some posts around there, and I saw this review. I said, hey, right, I never got around to doing that as a pick for TWiV. This is a book called A Paradise Built in Hell by Rebecca Solnit. And it's the book came out in 2008, I think. And I'm also linking the review that I put on my blog in December of 2020. So Solnit is a kind of a freelance academic who just investigates interesting stuff. And she got interested in disaster response. You know, big topic, what happens when the lights go out, when the earthquake hits, when the hurricane blows through. And what she found was that there's a whole sociology of disasters that is not what you see in the Hollywood version. So if you see any disaster movie, you know, the aliens attack, the zombies, whatever, or if it's, you know, climate disaster and there's some big weather event, you've got people running around in the streets shooting each other and looting stores, and it's chaos and everybody against everybody. But then if you actually look at disasters, and I think most people have been through at least one significant disaster. The lights went out for a week when we had an ice storm here. You know, I grew up in an area where we got hurricanes. People in California have had earthquakes. So you've experienced probably some level of a disaster. And probably it wasn't like the movie. In fact, what usually happens is that people go, you know, they come out of their houses and see, is it safe to get out of here? And then they start checking on their neighbors and seeing if people are okay. And they scrounge up supplies and, hey, you know, my freezer's out because of the power. I'm going to grill up all this meat. Everybody, you know, come on over and eat. So it's this huge actual outpouring of community support that is what we see in disasters. That's what really happens. And this book delves into, I think it's 4 different North American disasters encompassing the 20th into the early 21st century, starting with the San Francisco earthquake of 1900, whenever that was, and ending with Hurricane Katrina in 2005. And every time, one of the worst parts about the disaster is that the government response presumes everybody's going to be in chaos and makes things worse because they start running around, you know, with militarized police trying to get control when in fact things were doing fine if they didn't do that. So it's a really good book. It'll make you think differently about disasters. And the reason I bring it up now is because I expect we're going to have more disasters in coming years. You know, it's worth paying attention to how the response plays out and thinking about it.
Was COVID a disaster?
Oh, sure.
Yeah.
Yeah.
I agree. Yeah, we lost power for,
you know,
for
you remember from the early days of the pandemic, there was sidewalk chalk art, you know, thanks to our first responders and essential workers and people in Italy singing to welcome the doctors home after their shifts and all this kind of stuff. It was, again, this outpouring of communal action and support. Which is not to say we should have more disasters because people are nicer, but you know, if we incorporated that into our planning, it would make things go a lot more smoothly.
We're going to have, as we said today, we're most likely going to have infectious disease disaster.
Yes.
And is really that's what it's going to take to get people together?
Maybe.
Well, the last one did not really bring us together.
Beyond that temporary initial
Yes.
phase, no.
True. Yeah. All right. Thank you. That's pretty cool.
That sounds
But
like an
the
interesting
reason
book.
it didn't bring us together was because the official response was so toxic,
Hmm.
I would argue.
Yeah, there are a lot of problems with it. Jolene, what do you have for us?
Well, I am finishing a semester of teaching virology to undergraduates here at Texas A&M, and as part of my course, I required my students to have 4 public communication projects where they would choose a subject themselves from a very broad list that was allowed to convey topics about virology to the general public, to a non-expert audience. And they had pretty much free creative license in how they would execute these projects. And they chose many different topics, from myths to just learning about viruses and conveying results from research reports. And they made videos and blogs and poems and all sorts of infographics to convey those messages to the public, and they had to communicate it to someone and tell me that they did that. So a roommate or a family member. And I allowed them to then opt in to publicly sharing their project, and we voted for People's Choice projects. And so I'm sharing the ones that opted in, and the top 3 are marked here on the link. There was a cat meme about virus attachment. That was a TikTok video. And then there was a book about Andrew Wakefield, The Tale of the Bad Scientist. It's a children's book. And then there was Contagious Love, which was all about HPV and how you can prevent it. And it was really a lot of fun.
Great.
I think this is a critical skill, communicating accurate scientific information to people who don't know about it. And so I had a lot of fun. I hope that the students did. Anyway, I'm sharing the ones that are public here for your enjoyment.
This is very cool.
This is awesome. That is wonderful.
Once upon a time, there was a man who wanted to be rich, so he faked an experiment to scratch this itch. So that's what you said before.
Yes.
Andrew Wakefield wasn't about religion. It was about money.
He might have had some religious angle, but he was in it for the Benjamins. Yeah.
That's very cool. My pick is the Vaccine Education Center at the Children's Hospital of Philadelphia. I've known about this site for a long time, and it was recently sent to me by Lise, who is a listener. And it's just a great collection of information about all sorts of vaccines, diseases, safety, history, schedule, immunity, and Paul Offit is involved in this. So I want people to see it and look at it and get some information from it, because this is not misinformation. This is truth. So we need to collect sites that are good, and this is one of them.
Right. It comes from the
resource.
Children's Hospital of Philadelphia, also known as CHOP.
CHOP.
And it's got, it's not just like general information, vaccines are good. It's got very specific stuff like a complete vaccine timetable, schedule, stuff that you might actually just want to look up because you're wondering if your kid is due for a vaccine or you are. Very handy.
They have a nice explanation of the human immune system and history of vaccines.
2 listener picks. One is from Elizabeth. Dear Doctors TWiV, I'm writing to you from Bainbridge Island, Washington, where today it's a perfect 20C. I've been a faithful and grateful listener since 2020. I just work in book publishing, but I'm deeply fascinated by science. Your discussions delight me, teach me, and fill me with wonder. I don't know what I would have done without you in 2020. Thank you for your lifeline of science communication then and now. I'm not sure if the Dance Your PhD contest has been picked before, but it's such a gem of a science arts crossover. I couldn't resist sending in this year's winners. See, especially the overall 2025 winner from the University of Helsinki. The organizer of the contest notes that this year's winners were all European scientists. This year, American scientists did not seem to be in the mood to dance.
I wonder why.
Lucky for the world, European scientists have doubled their creativity and enthusiasm. I hope so. And so Elizabeth provides links, the one without a paywall in Ars Technica. Many thanks for all that you do. So it's if you taught PhD students, Jolene, you could have them make a dance.
I do. I have a dancer actually I have an actual ballet dancer and another person who
Yeah.
really likes to dance.
I've had in the past ballet dancers here in my course as well, and I always thought of doing something, and they were very enthusiastic about it. But
I got an interpretive dance as one of my communication projects, but they did not choose to submit it for the public voting.
Oh,
and from Blue Pilgrim in Eugene, Oregon, where spring is in progress and the weather is good. Upton Sinclair, born 1878, American novelist and social reformer. It's a quote from him. It is difficult to get a person, I'm going to change it from man to person. It is difficult to get a person to understand something when their salary depends upon them not understanding it. It's difficult to get a person to understand something when their salary depends on them not understanding it.
Indeed.
That's great.
Yeah.
And he said he gives a link to a quote site where you can find variations on it as well. Yep, that's true. Any example you could cite in the last 100 days is this. That's TWiV 1215. You can find show notes at microbe.tv/TWiV. You can send your comments, questions, pics to TWiV@microbe.tv. And remember, please support us financially. Microbe.tv/contribute. Alan Dove is at alandove.com and turbidplaque.com. Thank you, Alan.
thank you. It's always a pleasure.
Jolene Ramsey's at Texas A&M and at ramseylab.vercel.app. Thank you, Jolene.
Thank you. It's fun, important topics today.
I'm Vincent Racaniello. You can find me at microbe.tv, and you can find Microbe TV on Blue Sky. That's Microbe TV with no dot, microbetv.bsky.social. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV. Ronald Jenkins for the music, and Jolene Ramsey for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.